Ventilation or breathing is controlled by a number of factors. Respiratory rate is the number of breaths taken each minute, with approximately 12 to 18 breaths per minute being the typical breathing rate for a healthy adult at rest. This rate is called eunia. Eunia is controlled by clusters of neurons in the medulla oblongata of the brainstem. These neurons are sensitive to changes in carbon dioxide or oxygen concentrations within the bloodstream, in addition to changes in blood pH. All of these factors will cause the brainstem to alter breathing rates in response. The dorsal respiratory group receives information from peripheral chemoreceptors that are located in the carotid arteries. These chemoreceptors sense CO2 concentrations in the blood flowing through those arteries. The dorsal respiratory group neurons in the medulla oblongata will then use that information to modify breathing rate and depth of breathing. If CO2 concentrations are high, the rate and depth of breathing is increased in order to expel excess carbon dioxide by exhalation. Removing carbon dioxide from the blood also has the effect of eliminating acids from the blood and making the blood slightly more alkaline as pH increases. Purposely hyperventilating or hyperventilating during panic attacks can cause too much carbon dioxide to be eliminated from the blood and make the blood dangerously alkaline as a result. On the other hand, if CO2 concentrations are low, breathing rate and depth of breathing decrease, giving time for CO2 concentrations to accumulate back to typical levels. CO2 is never completely eliminated from the bloodstream. Instead, its concentration is held within a specific homeostatic level. Neurons in the ventral respiratory group of the medulla oblongata function to set eunia and control the diaphragm and external intercostals that establish quiet inspiration and quiet expiration. In addition, the ventral respiratory group also controls other skeletal muscles involved in forced inspiration and forced expiration.